Hello everyone. In the previous video, we have talked of the bacterial nutrition and its types. Now, in this video, let's discuss about the reproduction of bacteria. Basically, the bacteria exhibits two types of reproduction. One is asexual reproduction and the other one is sexual reproduction. Let's begin with the asexual reproduction. Asexual reproduction is a process where the new offsprings or the new individuals are produced by a single parent, and here there is no fusion of gametes or no formation of gametes. Only the single parent itself is producing the two offsprings, and the two offsprings which are produced are identical to each other. They are pretty much similar to each other. This is called asexual reproduction. And there are many types of asexual reproduction like binary fission, spore formation, fragmentation, and budding. But the bacteria which we are talking of is exhibits either the binary fission or the spore formation. They exhibit binary fission under favorable conditions. What are favorable conditions? Like the pH or the temperature, which are favorable for the bacteria to grow, and if there is enough food available for the bacteria to eat. Those are the favorable conditions. Under such favorable conditions, bacteria exhibit binary fission, whereas under unfavorable conditions, like if the temperature changes, pH changes, and if there is no food left for the bacteria to eat, under such extreme conditions, under such unfavorable conditions, bacteria exhibit spore formation. So let's see how the binary fission takes place. Yes. So this is the parental cell. This is the bacterial cell. Here, this is the genetic material of the bacterial cell. This is the cell wall, and this is the plasma membrane of the bacterial cell. And these are the mesosomes of the bacterial cell. These mesosomes are nothing but the enfoldings of the plasma membrane. So, this bacterial cell during the binary fission, their genetic material will replicate. So, here you can see the replicated genetic material. Once the genetic material is replicated, then of course the cell size will also increases. The cell size will enlarge, and the replicated genetic material will move to the extreme ends. They move to the uh, two poles, and they attaches to the mesosomes of the plasma membrane. If we recall our previous video, we have said that mesosomes are useful during the cell division, and this is how they work. These two genetic material will move to the corners, and they attaches to the mesosomes. Now, once the genetic materials are attached to the mesosomes, then the cell wall formation takes place. Once a cell wall is formed, then the cell slowly starts dividing, and it will completely divides and produce two individual daughter cells. So this is the concept of binary fission, and this whole concept of binary fission takes 20 minutes time to complete. See here, this is the single parent cell, and this parental cell will take 20 minutes time to produce two individuals, two offsprings, and this 20 minutes time is called as a generation time. If you take 100 bacteria, within 20 minutes they will replicate and form 200 bacteria. So this is about the binary fission. And also important thing here is this binary fission takes place under favorable conditions only. But what happens during the unfavorable conditions? How the bacteria survives during unfavorable conditions? See, during unfavorable conditions means if the temperature falls suddenly to minus 100 degrees centigrade, or sometimes temperature raises to plus 100 degrees centigrade, sometimes pH changes, and sometimes there will be no food available for the bacteria to eat. Under such extreme conditions, um, the most of the bacteria dies, but some bacteria produce endospore inside its body. So if you see, this is a bacterial cell, and this is the genetic material, this is the cytoplasm, and this green color layer is called as protective layer. And here, this black colored layer, if you see, this is the endospore. It is called as endospore because endo means inside. Since the spore is forming inside, it is called as endospore. And this is how the spore formation takes place. And mostly bacillus genus bacteria and the clostridium bacteria forms this endospore. Here is the bacillus bacteria which is having a rod shape, and here the endospore formation. Yes, this is how the spore formation takes place. And during once the unfavorable conditions are gone, when the favorable conditions are arrived, in that condition, this bacteria comes out from this endospore and again undergoes the binary fission. Okay, this is how the bacterial cell undergoes the asexual reproduction. Let's see sexual reproduction. When we hear this word sexual reproduction, we usually recall about gametes and gametic fusion. But in case of prokaryotes, in case of bacteria, there is no such gametes here. But some primitive method of DNA transfer occurs here. That means the DNA transfer from one bacterial cell to another bacterial cell. There are three types of sexual reproduction in case of bacteria. They are transformation, conjugation, and transduction. So let us begin with the transformation. Transformation is a type of sexual reproduction in which the bacterial cell takes up the foreign DNA and incorporates that foreign DNA into its own genetic material. Let's say this is a bacterial cell and it is having its own genetic material, and there is, let us assume this external space as the external environment, and there is a piece of foreign DNA present here in this external environment. This may be released from some other bacteria. Now what happens, this bacteria takes up this foreign DNA and incorporates that foreign DNA into its own genetic material. See here, the foreign DNA is entering into the cell and it entered here. Once it entered, it incorporates into the host genetic material. So once this incorporation has done, then this bacteria undergoes the binary fission. During the binary fission, this incorporated genetic material along with the foreign DNA replicates and pass on to the next generation bacterial cells, the new offsprings. So these two newly produced bacterial cells are having their own parental DNA along with this foreign DNA. Because of this foreign DNA, they express some new characters. Let's say this foreign DNA having antigenic resistance gene. So if since this foreign DNA is having antigenic resistance gene, these bacterial cells are also showing antigenic resistant characters. So this is all about the transformation. So here the main thing we have to remember is the bacterial cell is taking up this foreign DNA and incorporating that DNA into its own genetic material. So here the external environment is acting as a medium for this transformation. So this is all about the transformation. The next type of sexual reproduction in case of bacteria is transduction. Here, in case of transduction, virus mediated DNA transfer occurs. That means here there is a virus which transfers the one bacterial cell DNA into another bacterial cell. So here this type of virus is called as bacteriophage because this virus is actually attacks the bacteria. That is why they are called as bacteriophage virus. And if you see the structure, it have a viral DNA inside its head. It is a capsid and it is the tail part. So this is the structure. And let's see the process of transduction. The first step involved in the transduction is attachment. So here this is the bacterial cell which is having its own DNA, and this is the viral cell. It is having a viral DNA. This is called as bacteriophage. This bacteriophage comes and attaches to this bacterial cell. The second step is injection. That means here this bacteriophage slowly injects its viral DNA into this bacterial cell. And then in the third step, the hydrolyzation of this bacterial DNA occurs. That means here this bacterial DNA undergoes the fragmentation. It broken down into some fragments. And in the fourth step, what happens is here this viral DNA which is injected is replicated here. Along with this, the body parts of this bacteriophage also synthesized. So here you can see the capsid and the tail part. So all those body parts are synthesized, and also the viral DNA has replicated here. So now in the fifth step, all these replicated viral DNA and the body parts are assembled and packed. So here you can see the body part, and it is having a viral DNA. This is one bacteriophage, newly produced bacteriophage, that is having its own viral DNA. It is also bacteriophage having its own viral DNA. But here, if you can see, this is a bacteriophage, but it is not having its own viral DNA. Here, this bacteriophage is having a bacterial this host genetic material inside. So what happens is, these genetic material fragments, this bacterial genetic material fragments, mistakenly assembled into this bacteriophage body. So once all these bacteriophages are accumulated, then they release an enzyme called lysine, and with the help of that lysine, they break the cell wall and they releases out. Once they releases out, they again goes and infect other bacteria. So here, this bacteriophage, which is having this bacterial DNA, is is infecting other new bacteria. While infecting other new bacteria, it injects the bacterial DNA into the new bacterial DNA. So now this old bacterial DNA and the new bacterial DNA both will incorporate each other. So both fuses each other. So this is called as transduction. Now what happens? This one bacterial DNA, this bacterial DNA is incorporated into the other bacterial DNA with the help of a virus called bacteriophage. This is all about the process transduction. Then comes the next type of sexual reproduction in the bacteria is conjugation. So here the conjugation occurs with the help of sex pili. That means, see here, there are two types of bacteria cells present. One is donor cell, because it is having plasmid. If the plasmid is present in the bacterial cell, then it is called as donor cell. And it is also called as F plus cell, because this plasmid have a gene called F gene. F gene means fertility gene. So because of this gene present in the plasmid, they produce a sex pili. But if the plasmid is absent, then F gene won't be there, right? So if the F gene is absent, then sex pili won't form. So in case of recipient cell, it is also called as F minus cell, because it lacks the plasmid. Since it lacks the plasmid, it couldn't have F gene, and if there is no F gene, then it couldn't produce the sex pili. So this is the donor cell and this is the recipient cell. So it forms a conjugation. Sorry, it forms a sex pili, right? Now what happens, with the help of that sex pili, this donor cell fuses with the recipient cell. It forms a conjugation tube between the donor cell and the recipient cell with the help of sex pili. Once a sex pili, this conjugation tube is formed, then this donor cell starts donating its plasmid to the recipient cell. So here if you can see, this F plus cell, this donor cell has donated its entire plasmid to the F minus cell. So once this F minus cell receives the plasmid, then it will become F plus cell. And once F plus cell donates its plasmid to the F minus cell, then it become F minus cell. So this is one type of conjugation where the plasmid transfers from one bacterial cell to another bacterial cell. So this is all about the conjugation. So we have done with the sexual and the asexual reproduction of bacteria. I hope this video helps for you. If you like this video, please like, share and subscribe to my channel. Thank you.